Huntington's disease is a genetic disorder that causes oxidative damage and a progressive breakdown of brain cells. In rat studies, the herb ashwagandha protected against oxidative stress and mitochondrial dysfunction, suggesting it could be useful in diseases such as Huntington's. Astaxanthin, the algae responsible for colouring salmon, shrimps and prawns, is a powerful antioxidant that easily crosses the blood-brain barrier. The neuroprotective properties of this molecule involve antioxidation, anti-inflammation and anti-apoptosis. This may help protect the brain from some of the negative effects of Huntington's disease. New research published in September's issue of the Journal of Human Molecular Genetics has shown that the green tea extract, EGCG, may interfere with the accumulation of proteins in the brain that cause Huntington's disease. Huntington's disease patients were administered a maximum daily dose of 1,200 milligrams of EGCG for 12 months, and results were compared to placebo. Results were measured using the Unified Huntington's Disease Rating, Cognition Composite Score of Stroop Test, Verbal Fluency Test, and Symbol Digit Modalities Tests. The results showed that EGCG improved cognition in Huntington's disease patients. Research from the Centre for Molecular Medicine and Therapeutics at the University of British Columbia in Canada have discovered that fasting and restricting food intake to only 6 hours in a 24-hour period could potentially help slow down the pace at which Huntington's disease progresses. Huntington's patients inherit a genetic mutation in the huntingtin gene, which causes production of a form of protein called MHTT. Scientists believe that this protein is largely responsible for fueling the progress of Huntington's disease. Previous research has shown that fasting may benefit people with other progressive neurological conditions, such as MS, so the team sought to determine whether such a strategy might benefit people with Huntington's disease. The researchers came to their findings by studying mouse models of Huntington's disease. They restricted the rodents' access to food, so the mice were only able to eat during the same 6-hour period every day, and they fasted for the remaining 18 hours. The results were very encouraging. The fasting triggered a process called autophagy, in which cells clean themselves, removing damaged and unnecessary components. In addition to intermittent fasting, the ketogenic diet has been shown to have positive effects on neurological disorders, so it may not hurt to try these options under the supervision of a doctor. In fact, more than one animal study has discovered a potential benefit of the ketogenic diet or intermittent fasting in delaying weight loss, managing glucose, and protecting neurons from injury. Patients who exercise tend to do better than those who don't. Huntington's patients should aim for a body mass index at the higher end of the healthy range, roughly between 24 and 26. Fisetin, strawberry extract. It acts as an antioxidant, increases GSH, maintains mitochondrial function in the presence of oxidative stress, has anti-inflammatory activity against microglial cells, and inhibits the activity of 5 lipogenases, signifying that fisetin causes reduction in the age-related decline in brain function. The Mayo Clinic, in their first trial on women, gave 20 milligrams per kg of body mass. For women, that's 1,500 milligrams per day. For men, the dose is a bit higher, at around 25 milligrams of fisetin per kg per day. One way to take this is take it for 1 week and then take a break for 3 weeks and then continue. Resveratrol is found naturally in small amounts in red wine and Japanese knotweed. A phase 3 clinical trial, expected to include 102 patients with Huntington's disease, is currently recruiting participants in France. A total of 51 patients will receive resveratrol daily for 1 year, and the remaining 51 will receive placebo. The primary outcome is the degree of caudate atrophy, thought to be a sensitive marker of Huntington's disease. Caudate atrophy is the wasting of the caudate nucleus, a part of the brain affected by Huntington's. The atrophy or wasting of the caudate will be measured at the beginning and at the end of the study. Secondary outcomes are a reduction in the progression of the disease as measured by the Unified Huntington's Disease Rating Scale, which measures motor control, cognitive symptoms, behavioral symptoms, independence, and a person's ability to function in day-to-day life. Commonly used doses of resveratrol are 500 milligrams per day. Mitochondrial dysfunction has been described as an early pathological mechanism delineating the selective neurodegeneration that occurs in Huntington's disease. A polyglutamate expansion disorder that largely affects the striatum and the cerebral cortex. Cold water swimming has been shown to improve mitochondrial function. The supplement PQQ, taken at 40 milligrams per day, is one of the few things that makes the body dump old damaged mitochondria and generate new ones. The research chemical SR 9009 also does this in doses of 50 to 100 milligrams sublingually per day. Many supplements also feed the mitochondria, such as D-ribose, which is the only sugar that lowers blood sugar instead of increasing it. Coenzyme Q10 also, when taken in doses of 200 to 300 milligrams per day. In a study published in the Journal of Neurobiology of Disease, they showed that male Huntington's disease mice had different proportions of various bacteria compared to their healthy siblings. Intriguingly, they also ate more food, but gained weight at a lower rate. As their guts became dysfunctional and excreted excess water in fecal matter, the first movement symptoms of the disease would appear. However, the researchers were unable to say at this stage whether the altered microbiome plays any role in the timing of the symptom onset. The best probiotic that I would recommend is the strain VSL3, also known as Vivomix, as it has 8 different bacteria at 450 billion live bacteria count per sachet and 180 publications worldwide to support its use. Research just published in the Journal of Behavioral Neuroscience provides evidence that adequate omega-3 acids are needed for healthy nervous systems. That could explain why low levels of omega-3 are associated with the information processing difficulties experienced by people with bipolar, obsessive-compulsive disorder, and attention deficit hyperactivity disorders, schizophrenia, Huntington's disease, and other illnesses also affect the nervous system. What's more, the research suggests that increasing dietary omega-3s may be a natural way to prevent and treat some of those conditions.